Hello, so this video is about pathogens. So this is my favorite topic to teach. So let's get started. So this is basically the VCA study design, the key knowledge. So this is what you need to know for your VCE. You need to know the role of a human immune system in detecting and responding to antigens, the nature of antigens and their sources, self and non-self, and cellular pathogens and non-cellular pathogens, so viruses and prions. So our learning goal for this video is for you to be able to list the various types of pathogens and explain how they cause disease. You'll know you've been successful at this if you can list various types of pathogens and describe how they cause diseases, and you can differentiate between cellular and non-cellular pathogens. Okay, so what I'd like you to do during this video is you need to make sure that you pause the video at the end of each slide, if required, to take notes. So if you've had time to take notes during the slide, then you don't need to do that, but it's important that you take some notes while watching this video. Don't rewrite the whole slide, but just summarize the information in dot points and enjoy the video. Okay, so what are pathogens? Pathogens can be cellular or non-cellular organisms that cause disease. Parasites. Parasites are a type of pathogen that complete their life cycle in another organism called a host. Now, a parasite is only considered pathogenic if they actually cause disease in the host, okay? If they do not cause disease, then they are not pathogenic. Some parasites infect two different hosts in sequence: the primary host, which is used for the adult stage, and the intermediate host used for the larval stage. Some parasites are transmitted to their host via another organism called a vector. Now, endoparasites live inside the organism, whereas ectoparasites attach to the outside of a host. So what I need you to do now is pause this video and summarize this slide. Don't write the whole thing. So what I'd like you to focus on is define what pathogens are. So you might need this first sentence. Then you need parasites, which are a type of pathogen that complete their lives in another organism called a host. Okay. You need primary host and secondary and intermediate host. You need to define vector, and you need the difference between an endoparasite and an ectoparasite. So dot points, just summarize the information. Pause the video now and then come back when you're ready to move on. Welcome back. So this slide here is about identifying the vector in this scenario. So what you've got here is a mosquito, and within the mosquito you've got those the malaria pathogen, the malaria causing pathogen. So if this mosquito here bites a person and transfer the pathogen across to the person in forms of sporozoids. The person inside the body of a person, the sporozoids multiply, okay, and up in the person's blood, okay. So then you have another mosquito that comes in and bites the same person, and now the pathogen is transferred to that mosquito, and that cycle repeats itself. So that mosquito would now go on and can infect new people. So here, which organism do you think is the pathogen? Is it the vector? Is it the person or is it the mosquito? Have a think about it. So yeah, it's the mosquito. The vector is the mosquito because the vector is carrying the pathogen and transferring it to the host. Okay, so the host is the person, the mosquito is the vector, and the pathogen is the malaria pathogen. All right, so here are two pictures. A bird here with some parasites on the outside attached to it. I think those are some kind of insect. And here you've got, those are actually bacteria inside the blood. So that's blood, and you can see it's been infected by some kind of a bacteria, okay? Now, which one of those is the ectoparasite and which one is the endoparasite? Have a think about it. So ectoparasites are those ones because they are on the outside, whereas endoparasites live on the inside. So that's an endoparasite, and this is ectoparasite. Disease. So a disease is defined as any condition that impairs the normal function of an organism. So there are several types of diseases. There are non-infectious disease, infectious disease, and contagious disease. So non-infectious disease are basically anything that is not transmitted from one person to another. Okay, usually those kind of diseases are hereditary or congenital, innate, or idiopathic. Idiopathic means that we don't really know what's the cause of a disease. Infectious disease are those that are transmitted from one person to another. Contagious disease are those that are transmitted by direct contact, okay? So again, pause the video and take some notes and return when you're ready. Welcome back. Okay, so now we're going to talk about the different types of pathogens. So here, again, there's a lot of information. You don't need to copy the whole slide. Just take whatever note you need. What we need to focus on here with the different types of pathogen is which one's cellular pathogen and which one are non-cellular pathogen. So we are going to start with bacteria. And a bacteria is a cellular pathogen. Here is a picture of what a bacteria would look like. As you already know, bacteria are prokaryotes. This means that they do not have membrane-bound organelles. Bacterial cells have plasmid DNA. So plasmid DNA is circular DNA, as you can see here. And we have circular DNA. It's not enclosed within a membrane, within a nucleus. Some are motile, some are not. So the ones that have flagella, this is the flagella, they are motile. If they don't have flagella, then they are non-motile. They have, some have a really thick cell wall made of peptidoglycan. So the cells that have a really thick cell wall are known as gram-positive, which means they stain blue with a gram stain. And some have a really complex multi-layered cell wall, and those ones are gram-negative, which means they stain pink. So that gram stain, it's the same stain, but they stain different colors. So it's the exact same stain, but the gram-positive look blue down the microscope, the gram-negative look pink down the microscope. Some bacteria have endospores. Now, endospores are little spores that forms on the inside of bacteria that allow the bacteria to survive harsh environments. Some bacteria are aerobes, so which means that they require oxygen. Some can live with or without oxygen, which means they are facultative anaerobe. And some can survive, cannot actually survive with oxygen, and they are obligate anaerobes. Again, pause the video, jot down a few notes, and then let's move on. So probably here what you need to focus on is the fact that bacteria are cellular, prokaryote, which means that they do not have membrane-bound organelles. You probably need this information about gram-positive and gram-negative bacteria and the fact that it's made up of the cell walls made up of peptidoglycans. So this slide here is showing the how different types of bacteria organize themselves differently and have different shapes. So over here you have the common shapes of bacteria. So you have bacillus, which are rods, coccus, which are spheres, and spirillus, which are like little spirals. You also have, it's not on here, but you also have Coccobacillus, which is sort of halfway between a rod and a sphere. So like an oval shape, if you like. Over here, you see the different organization of those bacteria. So if it's a coccus, so spheres, if they are in pairs, then it's usually called a diplococci. If they're singular, it's micrococci. If they form chains, then it's streptococci. And if they form clusters, they are staphylococci. Over here, you can see some have flagella, some have endospores. Then again, you don't really need to know all of this in detail, okay? So, but it's just something that you just need to be aware of, but you don't really need to learn this slide by heart. You just need to know that there's different shapes of bacteria and different arrangement. All right, the next organism we are going to look at are protozoan. Protozoan are cellular pathogens, which mean they are cells and they are alive. They are actually eukaryotes, which mean that they have membrane-bound organelles. So a few examples of protozoan that cause disease are sporozoans. So that's what causes malaria. Okay, what they do is they can detect movement and they are attracted to movement. That's possibly how they actually make their way inside the mosquitoes, because they can feel when mosquitoes are moving and they move towards it. Okay, and they infect the mosquitoes, but the mosquito is the vector. Okay. Then you have over here sarcodinians, which engulf red blood cells and cause amoebic dysentery. So again, you see that's eukaryotes because they are membrane-bound organelles. And what that one does is it basically eats red blood cells and makes you really sick. Over here you have and what that does, it's got flagella, so it can actually move and make its way up your digestive system. It's got an adhesive disc that allows them to attach to the intestinal villi and cause diarrhea. They're pretty nasty. That is still a protozoan. The next one is Protista. Now with Protista, it's another cellular pathogen. That's what they look like. They are sort of halfway between a plant and a fungi. They are very similar to fungi in the sense that they produce spores and they organize themselves similarly to the way fungi do. However, they have cellular cell wall, which is what plants have, instead of a chitin cell wall. Chitin cell walls usually found in fungi. So some of them are motile as well and have a flagella, while some don't. So it's not quite a fungi, it's not quite a plant, it's something in between, which is called Protista. One example of a Protista is homicides, which causes mildew on leaves. So if you ever see something like that in your backyard, that's most likely caused by a Protista. Fungi. Fungi are cellular pathogens as well. They form spores, they have a chitin cell wall, they even have antibacterial properties. So often you'll notice that if you have mold growing on a plate, then bacteria will not grow near them because they have antibacterial properties. In fact, a lot of antibiotics are made from fungi. So from chemicals that comes from fungi. Not all fungi are pathogenic to animals. Some are not. One example of a pathogenic fungi is the Aspergilloma, which basically infect the lungs and cause issues with the lungs. Worms. Worms are actually multicellular. Okay, so this means that they are not made up of only one cell, but they have a whole system of differentiated cells. There are two types of worms, platyhelminths. They are known as flatworms. They are endoparasites, which mean that they are on the inside of the body. They live in the guts and they have a head that is attached to the walls of the gut. They have a neck that grows out by means of asexual reproduction. And they have a chain of segments that consists of the male and female reproductive organs. So those male and female reproductive organs can actually fertilize each other, and this forms a bag of eggs, which is then passed in feces. So the offspring do not stay in the host, okay? They get passed out. Then we have the second type, which is the nematode. Those ones are roundworms, hookworms, threadworms, pinworms, and they are present in huge numbers in the environment. They reproduce by sexual reproduction. However, the offspring do not leave the host. They stay inside the host and make the host really, really sick. They even have a complete digestive system, and then they can survive inside the host. Arthropods. Arthropods are multicellular organisms. They are usually transmitted by insect vectors, such as mosquitoes and fleas. Examples of arthropods are ticks, mites, head lice, body lice, and crab lice. Okay, now we're moving on to the non-cellular pathogens. All the pathogens we've talked about before were cellular. Now we're moving on to non-cellular pathogens. Okay, the main one, the most common one that makes most people sick are viruses. Viruses are non-cellular agents, and they can basically infect any type of cell. However, they are specific, okay? They can only infect certain types of cell. They are obligate intracellular parasites that cannot replicate without a host cell. This is because they do not possess the machinery required to reproduce. For example, viruses do not have any organelles, they do not have any ribosomes, they don't have the rough endoplasmic reticulum. This means that they can't actually produce any protein, and they can't actually reproduce by themselves. What they have, though, is they have their own genetic material, okay, which is protected by a protein capsid. There are certain types of viruses that can infect bacteria, and those are called bacteriophage. There are two types of viruses. There are naked viruses, which is basically only the nucleic acid, okay, so the genetic material, which is protected by a protein capsid. Then you have enveloped viruses, which have nucleic acid protected by the protein capsid, but it's found in a phospholipid envelope, okay? Usually the phospholipid envelope comes from the host cell. Okay, so here are the virus structures. The genetic material can be double-stranded DNA, RNA, single-stranded DNA, or double-stranded RNA, depending on what type of virus it is. Here is a picture of a bacteriophage. They have a head with the DNA in it. They have a tail, and that's where they attach to the bacteria. That's an animal virus. They usually look like that. So you've got the DNA on the inside and the capsid on the outside. Viruses life cycle. Viruses can only infect specific cells that have a receptor molecule that match the viral attachment protein. So this here is really, really important. It is very likely that you're going to have a question like that on an exam, very likely. So viruses cannot just infect any cell, okay? They will only infect cells that have a receptor molecule that match the viral attachment. So usually on the virus, those projections you see there, that's where the viral would attach to the cell. Now the cell needs to have a receptor molecule that match the viral attachment proteins. Those projections here are the viral attachment proteins. If a virus cannot attach to the cell, then they can't infect that cell, okay? This is because the virus needs to attach itself to the cell so that it can transfer its genetic material inside the cell. That's how it infects the host cell, basically. Some viruses, however, do not have the attachment proteins, and they have other mechanisms of infecting the cells, okay? So for example, some viruses are engulfed by macrophage. So a macrophage is a type of white blood cell that fight pathogens by engulfing them. They eat the pathogens. So when a macrophage eats those viruses, that's how the virus ends up inside the macrophage, and then the virus infects the macrophage, which is not a good thing, because that means that we can escape the immune system. We can hide inside macrophage. All right, viruses life cycle. So what the virus do is they'll attach to a cell. They'll attach to a cell. The genetic material enters the cell, but the protein envelope, the capsid protein, and the envelope remains on the outside. When the DNA or the genetic material is inside the cell, it merges with the cell's own DNA inside the nucleus or inside the mitochondria. It then uses the cell's metabolic machinery to reproduce and produce viral protein, which is what you see here. And then they reorganize themselves into new little viruses or virions. Then there's two things that can happen in the end. So either the virus cell would lyse the cell, okay, and escape, or they'll form buds and escape, okay? So that's another picture showing you the virus life cycle. Feel free to pause the video if you need to have a better look at it. I'm just going to move on. Okay, so this is another quite important thing to know. There's two types of viruses. There's lytic viruses and lysogenic viruses. The difference between the two is that lytic viruses actually lyse the cell when they infect neighboring cells, whereas lysogenic viruses do not lyse the cell. So after the virus have divided, so this here over here, that's an example of a lytic virus. After the virus has infected the cell and it's divided and multiplied, when it's ready to infect other cells, what they do is they break the cell open, they rupture the cell, and they lyse the cell membrane, and they escape. And they then go and infect neighboring cells. Those are lytic viruses because they actually lyse the cell to escape. With lysogenic viruses, what they do is after they've multiplied and made several copies of themselves, they actually form a vesicle with the cell's own cell membrane, okay? And they can escape by exocytosis, okay? This is exocytosis. But what happens in the process, because they have that cell membrane from the host cell around them, it now makes it hard for the host immune system to actually fight those viruses, okay? Because they've taken on the host cell recognition molecules. They've stolen the cell's recognition molecules, so now it makes it hard for the cells, for the immune system to actually detect the viruses, the lysogenic viruses. Okay, so moving on, so that's still viruses' life cycle. There's a few different types when it comes down to what type of nucleic acid they've got. So some viruses have got DNA and RNA viruses, or DNA and RNA viruses. So DNA viruses, they basically become part of a genome of their host cell, because their host cell, which are human or animal cells, have DNA as well. So if it's a DNA virus, it will just become part of a genome. If it's an RNA virus, it doesn't need to become part of a genome. The RNA can just go straight onto the ribosomes and start producing proteins. Then there's another type of virus called retroviruses, which have RNA. In retroviruses, they have an enzyme called reverse transcriptase. So what retroviruses do is their RNA is transformed into DNA using that enzyme. Then that DNA becomes part of a cell's genome. Viroids. Okay, so viroids is another type of non-cellular pathogen. They are basically tiny circular single-stranded RNA molecules, which are about 1/10 the size of the smallest virus. And they do not have any capsid or envelope. They're basically just little pieces of RNA molecules. They usually infect susceptible cells. So they are known as opportunistic pathogens. Opportunistic pathogens are basically pathogens that do not make you sick unless you are susceptible. Okay, if you're immunocompromised, then they'll infect you. That's an opportunistic pathogen. So far, those viroids have been identified to cause severe disease in plants. Prions. Prions are also non-cellular. Prions basically consist of proteins called prion protein, PRP, and they do not have any nucleic acid. Some prions are pathogenic while some aren't. So some PRP are normal and others are disease-causing, and they are known as scrappy PRP. Prions only infect cells that can replicate and code for new proteins. So basically any animal cell can do that. Any animal cell can replicate and code for new protein. So prions can then infect those type of cells. What happens is the amino acids of normal and disease-causing PRPs are really similar, okay? The only difference is they have a slightly different shape, okay? When normal PRP come into contact with disease PRP, the shape of a normal one changes and it becomes exactly like the shape of a disease PRP, okay? So what happens in infected animals, scrappy PRP accumulates in the nervous system and slowly deteriorates the nervous system. Examples of diseases that are caused by prions are Kuru in humans, mad cow disease, and scrappy in sheep. Okay, so next we'll be looking at defending against pathogens. That would be it for today. I hope you've enjoyed this video. Feel free to go back to it if you need to, and make sure you write down some notes in your books. Thank you. I'll talk to you next time.